DNA Technology in Nature with
Alexis.
Meriden.
James.
And Sadie. DNA, or deoxyribonucleic acid, is also known as the genetic code of life and is what makes up the way any living organism looks, differs what defects or diseases we may be prone to, and even the way we develop and reproduce. But what if there's something or some way that could change all of that?
Well, shockingly, there is and has been for the thousands of years. Before upgrading to what humans have developed today, humans primarily used a tactic known as selective breeding to increase the likelihood of traits of plants and animals that could be beneficial to the human population, or honestly, just for fun, like the glowing zebrafish.
This tactic is still used widely today, like in the case of dog breeders making the perfect little puppy, or in the case of bananas being bred that aren't as small in size. However, the way of modifying DNA is not quite as efficient as recent breakthroughs have shown.
Within the last 20 years, there have been serious developments in technology that have made the ability to edit DNA very simple, inexpensive, and efficient.
Now entering the ring is our new opponent, CRISPR, or Clustered Regulatory Interspaced Short Palindromic Repeats. That was a mouthful. Which took the world by storm, not only cutting the time it took to run experiments down by months, but also cutting the price down by nearly 99%.
CRISPR comes from a defense mechanism in bacteria, originally identified in E. coli, described as a RNA-guided immune system to fight off viral infections, and works as if it were a DNA archive, where virus DNA is integrated into the original DNA after an attack.
If and when the virus attacks again, an RNA copy is made from the archive and loaded into a protein known as Cas9, so that it can act as scissors and take out the virus's DNA, rendering its attack ineffective.
Scientists have since discovered the programmability of the CRISPR system and found that one can edit live cells by simply giving it a copy of DNA you wish to modify and inserting it. This opens up a whole new gateway for curing diseases like cancer and HIV, creating targeted medication, and even alter human life as we know it.
Within the past year, 2023, one way scientists have attempted to use CRISPR to improve the quality of life for humans is by breeding popular trees and reducing their lignin levels, producing a more sustainable forest and more wood production.
Rodolphe Barangot, CRISPR founder, and Jack Wang, tree geneticist, led the project in lowering lignin levels and increasing carbohydrate to lignin ratios, and eventually launched a startup called TreeCo based on their attempts.
They used a machine learning model and tinkered with potential 70,000 gene editing strategies. Their model concluded that 21 total genes affected the production of lignin levels.
Some of the benefits that could come from the success of their business or tree editing as a whole is the obvious ability to provide a more sustainable forest, and as Berenjo stated, CRISPR systems provide the flexibility to edit more than just single genes or gene families, allowing for the greater improvement to wood properties.
Lowering lignin levels could enhance wood characteristics such as strength, flexibility, and growth rate, and cut out a large process of paper manufacturing known as pulping, causing a faster rate in paper production. Additionally, it has an indirect effect on our environment, as the widespread adoption of these trees could create less overall carbon emissions to the environment, therefore theoretically making our environment less susceptible to climate change, etc.
There are also some negative outcomes that could happen with gene editing. The gene could create a mutation that is harmful to the environment and pass down to the next generation. Lignin is used to help the cell wall maintain its strength. Without the cell wall strength in these trees, pathogens, pests, and other environmental effects are more easily able to break down the tree.
Additionally, there are many other concerns about the ethics of DNA editing. Many are concerned about humans playing the role of God and changing the DNA of organisms we so choose for economic and ecological advantages. Should we try to combat these issues in another fashion, one that doesn't involve gene editing? What do you think?
Thank you for watching.
Woo!